Morning.
Morning, ma'am.
Murag bag-ong mata.
Medyo.
Good
Hi,
morning, good
Ivy. Good morning.
morning, Ives.
Morning po.
From Washington University in St. Louis. His research interests include population genetics as well as genomic and molecular evolution. Now, without further ado, let's give it up to moderator for today, Mr. Carlo Lapid. Sir Carlo, take it away.
Hello, everyone. Thank you very much for inviting me to moderate the Q&A for this webinar. It's really a pleasure and an honor to be here. Now, allow me to introduce our guest speaker for today. Mr. Frank Ambrosio completed his Bachelor of Science degree at Binghamton University in New York before starting his career at Cold Spring Harbor Laboratories. From there, he moved to Atlanta to accept a position at the Centers for Disease Control, where he worked in the Division of Tuberculosis Elimination, the Biodefense Research and Development Laboratory, the Division of Bacterial Diseases, and the Division of HIV/AIDS Prevention. At the onset of the SARS-CoV-2 pandemic, he was deployed to the CDC's International Task Force and then to the Technical Outreach and Assistance for States team. And now, as a member of the 𝗘𝘃𝗃𝗁𝗃 team, Frank serves the public health community as a bioinformatics scientist who develops cloud-native workflows, orchestrates international training series, and provides technical support to the collaborators around the world. Let's all welcome for his talk on the evolution of public health pathogen genomics, Mr. Frank Ambrosio.
Thank you, Carlo. Hello, everyone. My name is Frank Ambrosio, bioinformatics scientist with 𝗘𝘃𝗃𝗁𝗃 Genomics, and today I will be talking about the evolution of public health pathogen genomics. Last week, you may have met my colleague, 𝗘𝘃𝗃𝗁𝗃, who is also a member of 𝗘𝘃𝗃𝗁𝗃 Genomics, and I'm going to start sharing screen now. So again, thank you very much for having me today. I would like to especially thank 𝗘𝘃𝗃𝗁𝗃 Mindanao for organizing this event. Everyone involved, thank you very much. It is an honor to be here and present for you today. So I'm going to be talking about the evolution of public health pathogen genomics, and I would like to start off with a motivation for why do we do public health pathogen genomics. Throughout human history, we have battled a number of pathogens, and public health bioinformatics allows us to glean unique insights to a number of pathogens, whether they be viruses, bacteria, or otherwise. Throughout human history, 𝗘𝗓𝗔 pestis has plagued mankind with a number of plagues. Hundreds of millions of humans have died due to plagues from 𝗘𝗓𝗔 pestis, including during the Black Death and the Justinian plague. The variola virus is the underlying cause of smallpox and has led to a number of plagues as well, including the Antonine plague and the Japanese smallpox epidemic of the 735 to 737. Tuberculosis is another pathogen that has claimed many human lives, and I personally worked at the Division of Tuberculosis Elimination at the Centers for Disease Control, where we developed many assays for assessing antimicrobial resistance within tuberculosis. Plasmodium is the root cause of malaria and has claimed a number of human lives as well. Yellow fever caused an epidemic in 1793, and influenza has had many pandemics throughout human history. In the 20th century, we experienced 3 pandemics caused by influenza. occurring in 1918, 1957, and 1968. And many of us will still remember the 2009 H1N1 pandemic, which had 60 million cases worldwide, almost 300,000 hospitalizations, and caused over 12,000 deaths. And of course, the current COVID-19 pandemic, which has caused over 100 million cases so far cumulatively, over 1 million deaths, and currently there are 20,000 individuals hospitalized with SARS-CoV-2. So why do we need bioinformatics? Bioinformatics helps us take effective measures against the spread of these pathogens and make informed public health decisions. So what are these public health measures? We can quarantine individuals who have been infected. We can use appropriate sanitation techniques based on the particular pathogen. We can design new vaccines to help combat the spread of these pathogens. And we can employ population scale public health measures such as genomic surveillance and outbreak investigations. Now, genomic surveillance involves assessing the landscape of genomes for a particular set of public health pathogens. We can discover new genes conferring antimicrobial resistance. We can discover new strains of a particular pathogen, and we can track the propagation of an outbreak through a population. Particularly with the SARS-CoV-2 pandemic, we track strains in real time. We track the spread of new lineages, such as the XBB recombinant lineage or the BQ1.1 lineage, which are currently taking over the landscape. We can also assess the effectiveness of our existing vaccines against these strains by sequencing their DNA and seeing the areas of the spike protein that our vaccine is designed to produce antibodies for. So there are many things that we can do with bioinformatics, and there are many reasons to continue to perform bioinformatics in a public health context. Now, bioinformatics has a very rich history. Pathogen genomics has a very rich history. And the science, the history of the science is fascinating and is a subject I love to talk about. So where does public health science start? There's room for debate here, but I would personally say that it starts with the discovery of animalcules or microorganisms by Antony van Leeuwenhoek in 1676, when he was looking through a microscope at some rainwater or lake water and saw protozoa and bacteria moving around. After that, the discovery of nuclear material by Friedrich Miescher in 1869 was a huge turning point in public health science. Many people believe that the discovery of DNA was achieved by Watson and Crick. However, the discovery of nuclear material actually occurred almost 100 years prior to that. So Friedrich Miescher, who very rarely receives credit for this, was the original discoverer of nuclear material in 1869. After that, the Hershey-Chase blender experiment is one of my favorite experiments in all of public health science history. This experiment occurred at Cold Spring Harbor Laboratories in New York. And in this experiment, bacteriophages were tagged with either a protein fluorescence or a nucleic acid fluorescence. And they were allowed to infect bacteria. Then these bacteria were placed in a blender where they were blended and allowed to recombine. And the radioactivity was then measured after collecting them in a centrifuge. And this experiment actually determined that nucleic acids were the fundamental genetic material that passes on traits from one generation to another. So this is one of the most important experiments in all of public health science, and it was performed by Alfred Hershey and Martha Chase. And then finally, the discovery of the double helix by James Watson, Francis Crick, and Rosalind Franklin in 1953 was, of course, a foundational moment in bioinformatics history. Now, with the real advent of DNA sequencing, we can get into how bioinformatics truly started. The first nucleotide sequence ever recorded was in 1973, with Walter Gilbert sequencing 24 base pairs of the DNA LAC operator. 24 base pairs, and this took 2 years to produce this sequence. Nowadays, we sequence many more bases than this, and we'll get into the details of that later, but suffice it to say that it took 2 years to sequence the first 24 base pairs. Later that decade, Fred Sanger produced Sanger sequencing in 1977. These Sanger sequencing uses audio radiograms, which are the measure of each nucleotide at a particular position, and these had to be read by hand at first, which was, of course, a painstakingly slow process. The first bacteriophage sequenced was 𝗘𝗃𝗁𝗃. Many of you will be familiar with 𝗘𝗃𝗁𝗃, as it is the genomic sequence that we spike into many Illumina sequencing experiments. And this was a 5.4 kilobase pair sequence, so 5,400 base pairs. The first bacterial genome was sequenced in 1995, and this was 𝗘𝗵𝗈 influenzae at 1.8 million base pairs. So now we've gone from 24 base pairs to 1.8 million base pairs in about 20 years. And this was also performed with Sanger sequencing. Sanger sequencing is a fantastic technology. It is foundational to bioinformatics. We owe a lot of credit to Fred Sanger for inventing this technology and introducing it to the world. And many of the early sequences in our sequence repositories were produced using Sanger sequencing. So if you're unfamiliar with Sanger sequencing, I will describe it a little bit right now. So the original methods were almost archaic to our modern bioinformatics and DNA sequencing methods. Essentially, an enzyme was used to clip DNA at each of the 4 base pairs, and then gel electrophoresis was performed, and you would see a line at each nucleotide, and you would go by hand and determine which nucleotide was at each position in the segment of DNA that you were trying to sequence. Later on, fluorescence was used, and machines could then read the fluorescence and determine the nucleotide sequence from the fluorescent sequences. After that, capillaries were introduced, and the ABI-310 single capillary machine was released. And if any of you are familiar with the ABI-310, congratulations, that is one of the most legendary DNA sequencers of all time. With this sequencer, sequences could be read by a laser causing fluorescence in a capillary, and this sequencer was used for a number of years to produce many sequences for a number of pathogens. So now we enter the next generation of public health bioinformatics. In the year 2000, next generation sequencing truly emerged on the scene. And next generation sequencing simply refers to the practice of sequencing by synthesis or using Illumina technology for sequencing. Now, Illumina was not the first company to invent next generation sequencing. There is some controversy around this, whether it was 𝗘𝗃𝗁𝗃 or Lynx Therapeutic Technologies. However, Nick McCook led the pioneer team at 𝗘𝗃𝗁𝗃, which produced some of the foundational technologies for next generation sequencing, otherwise called massively parallel signature sequencing. Both of these companies were bought by Illumina, which remains the pioneer in next generation sequencing. The 454 Life Sciences technology, or the Roche GS20 machine, was released by 454 Life Sciences, and this was the first commercially available next generation sequencing platform. And this could produce 20 million base pairs in a single run. So now we have gone from the first 24 base pairs, which took 2 years, to 20 million base pairs in a single run. So this is an unbelievable advent in DNA sequencing. we have seen tremendous strides being made in the amount of data that we could produce in a single sequencing run. And this is important to understand because the technology that we use to analyze DNA sequences must parallel the amount of sequencing data we can produce. We must use more powerful sequencing analysis technology to keep up with the amount of data that we are producing. So next generation sequencing emerged in the early 2000s. In 2008, James Watson of Watson and Crick fame was handed his own genome on a hard drive. And this took $1 million to sequence his genome. But I mean, less than 55 years after the discovery of the double helix, one of the discoverers was handed his complete 3 billion base pair genome on a hard drive. So I say this to highlight how quickly the amount of data that we can produce on a sequencer has increased. The cost of sequencing a human genome has also decreased precipitously. The first human genome cost nearly $100 million. Nowadays, you can have a human genome sequenced for under $200 by a company like 𝗣𝘂𝗃𝗃𝗁𝗃. And in 2008, 2009, third generation or long read sequencers emerged. Now, these are not the cheapest sequencers out there, and they are certainly not the most high throughput sequencers out there. The most high throughput sequencer to date remains the 𝗘𝗃𝗃𝗁𝗃. The particular model that is most high throughput is the 𝗘𝗃𝗃𝗁𝗃+, which Illumina claims can sequence 128 human genomes in a single run. And the human genome at about 3 billion base pairs per genome, you're looking at close to 500 billion base pairs per run. So again, we started at 24 base pairs in 2 years, and now we are looking at nearly 500 billion base pairs in a single run. So needless to say, the compute technology required must also increase. Now, this next slide is almost a required slide in any talk that concerns human genome sequencing or pathogen genomics in any way. The cost per human genome, which is the benchmark for how much sequencing data one can produce in a single sequencing run, has decreased at Moore's law for the first few years. And then we see in about 2007, there was a significant drop off with the release of the Illumina MiSeq instrument. Beyond that, the HiSeq instrument was released in 2014, which led to another decrease in sequencing cost. And these sequencers, they don't just sequence human genomes. Of course, they sequence many pathogens that we talked about earlier. And especially right now, SARS-CoV-2 is the most sequenced pathogen on the planet. We have never seen so much sequencing occur in such a short period of time. And this means we must adapt our compute platforms accordingly. And we will get into that in just a second. One more bit of history for all of us. This timeline here illustrates when many of the model pathogens or model organisms rather were sequenced. And so starting in 1960s, 1970s, we see 76 base pairs. We see E. coli 5S RNA at 120 base pairs. Moving on to when 𝗘𝗃𝗃𝗁𝗃 was sequenced in 1977 at 5.4 kilo base pairs, so 5,400 base pairs. Beyond that, we can see Haemophilus influenza in 1995 at 1.8 million base pairs. And then the human genome in 2004 at 3 billion base pairs. Beyond that, many other model organisms were sequenced, but at that point, we had shown that we can sequence the human genome, meaning we can sequence any bacterial or viral genome. Now it really comes down to throughput. How many sequences can we analyze? How much can we do in one run? And here we introduce bioinformatics. So how did bioinformatics initially work? Well, it was originally done by hand. As I showed earlier, we had to interpret the gel electrophoresis slides by hand and determine the sequence one at a time, one nucleotide at a time. And this was a painstakingly slow process, and we did not have very much that we could do with the sequence aside from compare it to other sequences, of which there were very few. Nowadays, we have enormous databases that we can compare all of our sequencing data against to determine where our sequences fit in the global context, in the global phylogenetic tree. And from that, we can glean information about new sequences as they come out. So bioinformatics, how did it start? Well, originally, bioinformatics was performed on a local computer, whether it be a desktop or a laptop. Scripts were written in Bash and Perl and C++, and they were run on the local terminal. You had the compute resources available at your fingertips, and it was not a lot of computing power. Oftentimes, you would see less than a gigabyte of RAM, less than a gigabyte hard drive, and it was very difficult to analyze more than 2 or 3 genomes from a bacterium. Algorithm development was especially important during the early days of bioinformatics. So we're talking the 1970s at this point. And of note, in the 1970s, we had several advents in alignment algorithms. Many of you will be familiar with the Needleman-Wunsch global alignment algorithm. Needleman-Wunsch was invented in 1970 by Saul Needleman and Christian Wunsch. And this algorithm is still used today for optimal global alignment. Now, a variation of the Needleman-Wunsch algorithm, known as the Smith-Waterman algorithm, was released in 1981, and this algorithm is also still used today. And this algorithm is best for local alignment. So rather than being optimal for global alignment, Smith-Waterman is best for local alignment. Now, the one alignment algorithm I know that everyone will have heard of is the BLAST algorithm. And the BLAST algorithm was originally published in 1990. It has had many, many version releases since then. And at this point, the original BLAST algorithm paper that was released in 1990 has been cited over 75,000 times. So it's a very popular alignment algorithm. It is used in many, many bioinformatic programs, including some that I have developed. So one of the biggest problems with performing bioinformatics on a local device is the limited amount of throughput. You can only process 1 or 2 samples at a time on a local computer unless it is very, very powerful. And with the advent of next generation sequencing, there was a tremendous amount of data available. So for example, if you are trying to analyze 1 bacterial genome at a time on a standard laptop with 8 gigabytes of RAM, it will take you roughly 2 or 3 hours just to process that genome, do a basic alignment, do some antimicrobial resistance testing, add it to a phylogenetic tree. If you try to do all of that on 1 laptop, now you're looking at 4, 5, 6 hours per genome. And if you have 96 genomes to analyze, you could be working on that 1 set of samples for over a week. So naturally, more compute power is required. So the early adaptations to increase the amount of compute power were the addition of high-performance compute clusters. And compute clusters are essentially many computers all linked together so that they can share resources and make the most out of whatever available resources are there. Now, HPCs, or high-performance compute clusters, they greatly increased the amount of throughput one could have, but they do require a great degree of technical expertise. You have to write very specific scripts to distribute the compute resources or distribute the jobs amongst the available compute resources. Now, many universities and public health organizations around the world will have an HPC, and this is still the state of the art for most of the world, including many laboratories that I worked in at the CDC made use of the high performance compute cluster on campus. And this is a fantastic advent in bioinformatics, but we are currently experiencing a revolution where we are moving beyond the era of the HPC into a new era where we are focusing on cloud compute technologies. Now, one advent that occurred simultaneously with high performance compute clusters emerging on the scene was the emergence of the Python programming language, which is my personal favorite programming language. I do not mean to cast aspersions on R or Perl or Bash or any of the others out there. They are all fantastic languages, but I do believe that Python makes programming more approachable for the beginner bioinformatician. So with the advent of Python and high performance compute clusters, we can now analyze many, many genomes. HPCs can really handle that NovaSeq level output where you have hundreds and hundreds of genomes and up to 500 billion base pairs being produced in one shot. The issue now is when you have multiple NovaSeqs all in one facility. And this becomes an issue, especially during the SARS-CoV-2 pandemic, when a large injection of funding for DNA sequencers is available, and many sequencers are purchased in rapid order. And all of a sudden, the amount of sequencing data being produced increases dramatically. And in order to scale your compute resources to meet the needs of your DNA sequencing technologies, it is very difficult if you have a local compute infrastructure. Managing a local compute infrastructure is a full-time job. And we as biologists and bioinformaticians and genomic epidemiologists, we're not in the business of compute infrastructure. We're scientists, we're biologists, we're geneticists. Compute infrastructure is best handled by professionals in compute infrastructure, in my opinion. So the big advent there is cloud providers. And for example, Azure, Google Cloud, AWS, they handle infrastructure management very well, meaning they have compute centers all over the world that are managed by individuals who are experts in compute infrastructure. They have redundancy built into their system. They have server software updates. They have hardware updates. They take care of the cybersecurity for you. All of this is handled by the cloud provider. Now, data backups and redundancy, I would not 100% rely on the cloud provider. For that, many people like to use a hybrid model where they have some data backed up locally and other data backed up in the cloud. And additionally, dynamic resource management is a huge advantage of working in the cloud, meaning when you have an HPC, you might have, let's say, for example, 100 computers in your high performance computer cluster. You might not be using all 100 computers all the time. You might only use all 100 computers when you have 10 NovaSeq runs all going at the same time. Now, with dynamic resource management in the cloud, you only use the amount of compute resources that you need. So if you only need one CPU, you only pay for one CPU at a time. You only utilize one CPU at a time. If you purchase an HPC and you have it on campus, on site, those resources are there no matter what. And that is still the state of the art, but we are seeing a slow shift towards cloud computing for all of the reasons I've laid out here. Now, taking care of all of the updates, the hardware updates, making sure everything is up to date as possible, it's a full-time job. And eventually, you will hit a threshold, a physical limit on the amount of compute power that you can pack into one high-performance computer. Now, I've seen an HPC in person. They can take up an entire floor of a building or an entire building in and of itself. And that physical space comes at a premium. You are paying for that physical space. So in my opinion, we are going to see a shift in the bioinformatics community moving forward from local high-performance compute clusters to cloud-based compute resource utilization. And I personally have started using many more cloud compute resources than local compute resources since starting with 𝗘𝘃𝗃𝗁. Now, the cloud has a lot of additional benefits outside of just the amount of compute resources available. Particularly in the field of bioinformatics, there are issues with developing workflows, sharing the data that comes out of a workflow, and just performing analysis. So in the cloud, collaboration is facilitated by the fact that multiple individuals can have access to the same resources at the same time. So for example, I can launch a workflow and I can monitor the status of that workflow in real time, but so can everyone on my team, whether they are on site or they are halfway across the country or halfway across the world. As long as they have access to my account, they can see exactly what is going on with a particular analytical workflow. The ease of access for end users. So oftentimes I write workflows that are designed for laboratorians to access. And these laboratorians will not necessarily be near me or on the same campus as me. So being able to provide access to my workflows to anyone, anywhere around the world is a huge advantage, especially nowadays. Sharing the results of my workflows with the team can often be a problem if you're just trying to send a file. As many of you will know, that bioinformatic files can be rather large. So sending a file in an email is often not an option. So the cloud-centric model allows you to, rather than sending a file, you simply point to a file. And this allows you to perform subsequent analyses on a file without making many, many copies of it. If you have a file on your computer, say it's a 𝗲𝗃𝗃𝗁 file, say it's a very large 𝗲𝗃𝗃𝗁 file that came out of a 𝗘𝗲𝗃, rather than copying that 𝗲𝗃𝗃𝗁 file to another person's computer so they can also analyze it, if that 𝗲𝗃𝗃𝗁 file exists in the cloud, many people can access the same file from the same location without making multiple copies of it. And we can all perform our different analyses on that same file. So that is a tremendous advantage. You end up Sorry about that. Check 1, 2. Is my microphone working? Okay.
We can hear you
Sorry
now.
about
Thank
that.
you. No problem.
But for the reproducibility of analyses, if you have a file of your code saved on the cloud, you can ensure that it is versioned and available everywhere so that individuals that you are collaborating with can access that same version of the software everywhere and anywhere. So reproducibility and version control are very important, especially if you're trying to collaborate with individuals on a project. You want to make sure that you are using the same bioinformatic pipeline as someone else without any of the dependencies being different versions. So version control and reproducibility are facilitated by cloud computing because you can access the same exact file instead of trying to share a file back and forth. Now, in the field of bioinformatics workflow development, I like to break it down into 3 steps. There is the development or the actual implementation of the bioinformatics analysis, the writing of the analytical code, and that's the first step here. The next step is the distribution. How do I share this code with someone else? How do I ensure that they can use it in the same way that I am using it? And operationalizing analytical software is a challenge, especially if you are working on different operating systems or if you have a completely different compute environment. And then finally, the production platform. So if I am collaborating with another individual to develop a bioinformatic pipeline, that's a challenge, but then the end user might have a third operating system or a third compute infrastructure that they are using for their analysis. And that can be particularly challenging, especially if you're dealing with the differences between Windows, Mac, Linux, all at the same time. And many of these issues can be addressed by using a suite of modern bioinformatics development tools. So let's go back to the beginning. How did things start? Well, development occurred on a local hard drive. And this is still the case in many, many laboratories around the world. And I have definitely developed on a local hard drive in my career many times. It's not bad practice by any means. It's just not optimal. So when you're developing on a local hard drive, you save a file and you can share that file with someone else. It might be the same version. It might be version 2_final_this one. I know that I have used that system of saving files with versioned file names before, and it is less than ideal. It's very easy to accidentally overwrite a file or save it with a file name that is too similar to another file name and get confused. Next up is the distribution. I have seen many times in my career files being shared via email or even being saved on a hard drive and physically handed to someone else so that they could load the file on their computer. Now, sharing files with no true version control system is dangerous because you don't actually know if you're using the exact same file as someone else. So when I write a workflow, I like to save it in GitHub or some other version control software where we can all be sure, all of my collaborators can be perfectly sure that we are all using the exact same workflow. Now, the production platform that you use is up to you. Many people nowadays like to use a graphical user interface or a codeless interface because it is difficult to learn to code. And oftentimes you will have specialists who are deeply entrenched in a field such as molecular biology, and learning to operate a workflow at the command line is outside the scope of their specialty. So we like to use graphical user interfaces nowadays, but back in the day, originally, we used the local terminal, which meant you had a command line prompt and you had to type in commands and pipe the outputs from one program into the inputs of another program. And this was, of course, not ideal. It led to many issues, many failed workflows, and it was very difficult to reproduce the exact same analysis if you did not have everything saved appropriately. Now, again, there were major improvements as we increased the amount of sequencing data we could produce. We had to move from a local computer to an on-site server. And a server is essentially just many computers all plugged in together. And this allowed for massive improvements in throughput. Where you used to be able to analyze one genome in a couple of hours, now you can analyze hundreds or thousands of genomes, even with your on-site server. And this is a tremendous development. This is still the state of the art today. You can handle enormous throughput, and it is a fantastic piece of technology, the on-site server. However, the downsides, it does require maintenance. Managing an on-site server requires a sysadmin typically. And the sysadmin, a system administrator, will perform the updates to the server operating system. It will perform the cybersecurity requirements on that server. And even then, even with a very powerful server, you may not be able to handle the amount of data that will be coming with the next big breakthrough in sequencing technology. The next big advent was in the distribution field. So in distribution, we moved from emailing files back and forth to using FTP systems, so file transfer protocol systems or secure file transfer protocol systems. And this allows us to send and receive very large files, so much, much more data than you can send in an email. You can send through an FTP. And this is still used by many laboratories and public health institutions around the world today. It's a fantastic technology. FTPs are great. That being said, you are still making copies of the file. So when I say you're making copies of the file, you are taking up space on multiple hard drives when you share this file. Whereas with a cloud-based system, the file exists in the cloud and you simply point to that file from other analytical pipelines. You don't need to make copies of it. And this ends up saving space across many different file systems. The next advent was in the production platform. So again, if you have used a local cluster as your production platform, that's fantastic. High performance compute clusters allow for tremendous throughput, but again, they do require maintenance. They require specialized scripts to distribute the jobs across the available resources on the compute cluster. And this can be limiting for bioinformaticians. Oftentimes, bioinformaticians want to focus on the analysis of DNA, not on how to distribute their analytical workflows across resources. And if you have to spend a lot of time figuring out how to operate SunGrid engine or another distribution engine, it can be very limiting in the amount of actual analysis time or the amount of time that you spend figuring out the actual analysis that you want to perform. So next up, we have the modern bioinformatics workflow deployment system. Now, this is the system that I use, so I am definitely biased here, and I will say that up front. Even if using a high performance compute cluster, you're going to face limitations, and I have faced those limitations in the past. So now I am fairly biased towards this setup, and I'll go through it step by step. So first up, we have the development step. I like to use GitHub as my code hosting platform. You don't have to use GitHub. There are many other open source code hosting platforms available that are just as good as GitHub, but GitHub is the largest, I believe. And for me, it's the most intuitive to figure out. It's particularly useful if you're doing open source code development. And open source code development simply means that you are sharing your code with the community. And to me, that's most important when you're doing public health pathogen genomics. I have the philosophy that if you are using public funding to write code and develop workflows, those workflows should then also be available to the public. And GitHub allows you to open source your code and make it available to the public health community. Now, there are many benefits from using GitHub. We'll go through those in just a minute. DocStore is the next advent. This distribution platform allows you to take containerized workflows and package them in what's called a WDL script or a workflow orchestration language script and distribute the entire workflow in one shot. So if you're unfamiliar with Docker containers, what a Docker container is it's like a small virtual machine. And the virtual machine, if you're unfamiliar with that, it's like a small computer that exists in the cloud somewhere, and it has an operating system. It has all of the dependencies required to run your workflow already installed. So you don't have to worry about installing the right version of each of the dependencies and getting your workflow to operate successfully. All of that will be pre-installed in the Docker container. So it's very easy to download a Docker container or download an image and spin it up and spin up a container and start doing the analysis and not worry about installation of software. Like I said earlier, as a bioinformatician, I want to spend most of my time focused on the actual analysis, not on the infrastructure and not on distributing my job across the compute resources, and definitely not on installing dependencies. And then next up is the production platform. So there's a lot of debate right now about whether or not graphical user interfaces are the way to go for bioinformatics software. And the reason for that is, while a graphical user interface makes the software easier to use for the end user, it does make it a bit more difficult to string together multiple bioinformatics tools if they're only available in a graphical user interface.
This is why I think the 𝗘𝗃blive platform is the perfect balance. 𝗘𝗃blive acts as the graphical user interface that Dockstore Docker container workflows can be plugged into on the backend. So you maintain the command line version of the tool while allowing a standardized graphical user interface that any bioinformatics platform or any bioinformatics workflow can be plugged into. So I like to think about it like this. 𝗘𝗃bio is the gaming console, and then your bioinformatics workflows are the games that you can plug into the gaming console. I guess games don't plug in anymore. They just download them. But back in the day, if you had a Sega Genesis, you would plug the game in, and the platform itself could accept any game. And that's exactly what 𝗘𝗃bio is. It can accept any bioinformatic workflow, and you have a graphical user interface immediately available for that workflow. And that's why I like using 𝗘𝗃bio, because all of my bioinformatic workflows now have a graphical user interface that is accessible from anywhere in the world, because it is a web-based application. So I don't need compute resources on my laptop at all. I can operate my workflows from my laptop or from a tablet or even from a cell phone. I do not need any compute resources because it's all being deployed to the cloud. So let's go through each of these in a little bit more detail. First up is GitHub. Everyone should know about GitHub by this point, but there's some really important aspects to operating or developing on GitHub that I want to highlight. First of all, version control is super important, making sure that you have every bit of software versioned. Not only allows you to reproduce the results that you produced with your bioinformatics workflow, but it allows others to benchmark your software. So you can do these large benchmarking studies and make sure that you're using the same version every time. The advent of development branches allows you to test out new features and grant access to these new features to beta testers or power users around the world. Whenever I add a new feature to a workflow, I do so on a development branch, which allows me to give instant access to anyone who wants to test out the new features in my workflow. And then once we've all tested it out, I can merge the development branches back into the main branch and add it to the main version of the workflow. Repository forks are an interesting concept in GitHub. Because GitHub is an open source code hosting platform, if you find a workflow out there in GitHub that you would like to create a new version of, you can fork a repository and add features or change the workflow to your delight and customize it for your particular needs. And this is fantastic. This allows everyone in the public health community to go on GitHub, find whatever workflow best suits their needs, and modify it to be exactly what they need for their particular vein of research. GitHub also allows for automated testing, which is one of my favorite parts about using GitHub. When I push a new commit to GitHub, or I push a new update to my code to GitHub, an automated test is triggered to ensure that this code will not break the pipeline. And that's fantastic because it avoids any code that would break the pipeline from making it to the main version of the pipeline, so that our collaborators do not end up with a broken version of the workflow. Aside from the automated tests, you can have manual code review on GitHub. So on GitHub, it's possible to tag one of your collaborators and say, hey, I have recently made updates to this pipeline. Would you mind checking them out and making sure that they look reasonable to you? And you can tag multiple individuals and they can leave comments and you can go back and forth. And in that way, it becomes this really beautiful open collaboration environment. And that to me is one of the best parts about GitHub, is the open collaboration. Anyone who wants to can review your code. and leave comments and submit an issue. If they find an issue, they can alert you of that issue. And in that way, you end up with this very rich history of the code development with many individuals from all different organizations contributing to your code base. And for me, that is one of the best parts about working on GitHub. And I am seeing some questions in the chat box. I will address these questions at the end. I just wanted to encourage everyone, please keep posting those questions. I do see them in the chat box. Thank you for the questions. I will address them at the end of the lecture today. Thank you. So the open collaboration in GitHub is definitely my favorite aspect. Another cool part about that open collaboration in GitHub is all of the comments and issues are tracked. So you can go back and see the history of a pipeline. You can see every glitch, every bug, everything that got fixed along the way, and everything it took to achieve that beautiful bioinformatics pipeline, the finished product. You can see everything that contributed towards that finished product. Next up is DocStore. So DocStore is where we host our workflows. And by workflows, I mean each of the components of a bioinformatics pipeline can exist in a script, in a Python script or a Perl script or a Bash script, but something needs to wrap them all together. And I'm sure you've heard the term wrapper script. Back in the day, we used to write Bash scripts that would wrap together many tools. Nowadays, we use workflow orchestration languages. For example, WDL, the workflow description language. Another example would be Nextflow, or SnakeMake is another example. And these languages, what they do is they allow you to take Dockerized components of a pipeline and string them together so that you can have multiple different tools that all require different dependencies. And rather than having to install all of the dependencies in one environment, you can have multiple different Docker containers that work together. So for example, if you have two tools and they both require Python, but one requires Python 2.7 and the other requires Python 3.7, that could be an issue if you have to resolve that in one environment. But with containers, that's not an issue at all. You have 2.7 in one container, 3.7 in the next container, and they are orchestrated by the WDL language. So this allows you to not worry about dependencies at all anymore. And for a beginner bioinformatician, installing dependencies is often a challenge. And I know for me personally, when I first started bioinformatics, getting all of the dependencies installed would give me nightmares. And nowadays, I don't need to worry about dependencies at all. So I'm very happy with Docker containers. And also the fact that each component of my workflow can be versioned is very nice for reproducibility. So if I want to swap out one component of my workflow, say, for example, I want to use Trim-O-Matic instead of FastP to trim my reads, I can just swap out the container at that step. I don't need to worry about breaking the pipeline or messing up any of the dependencies. I just swap out that container and set the new container in place, and the new pipeline is ready to go. And I can save that version of the pipeline and upload it to DocStore under my organization, the 𝗘𝘃cie𝗹𝘂𝘁𝗶 organization, and it's available to everyone. Now, DocStore is one of many scalable workflow repositories out there. One of the nice parts about working with DocStore and 𝗘𝘃cie𝗹𝘂𝘁𝗶 is that DocStore and 𝗘𝘃 are integrated, meaning when you go to DocStore, if you go to the page where my workflow is hosted, there is a button that says Open in 𝗘𝘃. And you click that button and you can immediately launch the workflow on the 𝗘𝘃.bio platform. It's very nice for making my workflows accessible to the public health community. Next up, we have the 𝗘𝘃.bio platform itself, and I've spoken about this a little bit. The 𝗘𝘃.bio platform is a graphical user interface bioinformatics platform that allows you to take any Dockerized workflow and run it with a graphical user interface. So every workflow I make, I have the same look and feel because 𝗘𝘃𝗃𝗁𝗃 provides the same look and feel for every bioinformatics workflow. I have inputs that are typed in. There's no code required. You don't need to type together different components. You simply open up the workflow, type in the inputs, and click Run Analysis. Now, in 𝗘𝘃𝗃𝗁𝗃, as opposed to many other workflow applications on the web, 𝗘𝘃𝗃𝗁𝗃 displays the outputs in a spreadsheet-like format. And to me, that is very, very important when you have a graphical user interface in the web, because you don't want to have to download the files in order to see the outputs. I want to be able to see the outputs while they're still up in the web without having to do any downloading. So 𝗘𝘃𝗃𝗁𝗃 allows you to see all of your outputs in a spreadsheet-like format. It also allows you to share access to those spreadsheets with your collaborators. So you can very easily just add someone's email to your workspace in Terra, and they will have access to all of your analytical outputs from all of your workflows. Also, Terra operates on the cloud. So all of your compute and storage is in cloud locations. So if you want to share data with someone, you're not making copies of that data. You are simply pointing to the Google bucket or cloud location where the data is stored. And of course, it is web-based, meaning if I decide to take my laptop and travel to another country, I can open my laptop and instantly have access to 𝗘𝘃𝗃𝗁𝗃, regardless of where I am. And this is a nice advent. I know with an HPC, you can SSH into an HPC from a remote location. However, with the web-based nature of this, I do not need SSH access. I simply go to 𝗘𝘃𝗃𝗁𝗃, log in with my Google credentials, and I immediately have access to all of my workflows and all of my data in the cloud. So today I have described a model to develop bioinformatics workflows in collaboration with the public health community. And when I say with the public health community, I literally mean everyone and anyone in the public health community can go to my GitHub and check out the code, contribute to the code, copy the code, fork the code, submit an issue or a bug report, submit a feature request or an enhancement request. And we collaborate with many different states around the country, many different countries around the world. And I believe last week, my colleague, Kevin Libowit, showed a couple of maps that showed how many collaborators we have all around the world. And it really is a community. I know that I might be the one pushing the buttons on the keyboard, but really all of the ideas come from everyone. The issues that arise in my code, they are discovered by my collaborators often. So I just want to give a shout out to everyone and anyone who has ever contributed to any sort of GitHub project. You are doing good work, and I appreciate you and thank you. So when you're working on GitHub, you have an open source development platform, which enables biologists and epidemiologists and scientists to contribute to pipelines that they will eventually use. And you don't need to know how to code to contribute to these pipelines. If you want to go on GitHub and contribute to a workflow that I'm developing, you can simply type in the feature that you would like to see added to the workflow, and I can start working on adding that feature. And in this way, it's, we get ideas from molecular biologists, from geneticists, from epidemiologists, people who don't necessarily know how to implement the code to perform the analysis, but they know what analysis needs to be performed. So we collaborate in that way, and it's a very nice collaboration. And it's facilitated by the use of that open source development platform. The scalable workflow repositories grant instant access to bioinformatics programmers around the world. So if you would like to use a containerized bioinformatics tool, you can access it. You can download the image and spin up a container and start using the tool immediately. There is no need to start installing dependencies, building an environment, as that can be very time consuming and complicated. And finally, the graphical user interface made accessible through 𝗘𝘃cie𝗶𝗂𝘁𝗵 allows scientists to focus on their analysis rather than learning how to write the code to perform the analysis. And that to me is something I'm very passionate about. My career started in the laboratory, and I would perform DNA sequencing, and then I would have to hand off all of my sequencing data to the bioinformatics team, and then they would send me the results back. And I wanted to be able to do the analysis myself. And now with the graphical user interface of 𝗘𝘃cie𝗶𝗂𝘁, I feel that I can empower laboratory scientists to perform their own analyses. So that makes me very happy as a bioinformatician, and I'm proud to be working with 𝗘𝘃cie𝗶𝗂𝘁. And with that, I just wanted to give some acknowledgements. First of all, the Broad Institute of MIT and Harvard for developing the 𝗘𝘃cie𝗶𝗂𝘁 platform, and specifically the 𝗘𝘃cie𝗶𝗂𝘁 team for being amazing collaborators and offering this tool to the public health community. The Staff B group, Staff B, which is not a public health pathogen, but rather an organization of state public health bioinformaticians who host a variety of Docker containers and various bioinformatic tools available to the public health community. The Public Health Alliance for Genomic Epidemiology, or 𝗘𝘃𝗃𝗁, as they're called, for making tremendous efforts to standardize workflow inputs and outputs and provide the public health community with a set of best practices. Next up is the Association of Public Health Laboratories for all of their good work, making connections between various public health institutions around the world. And last but not least, the entire 𝗘𝘃𝗁𝗁 genomics team who I work with, and without whom I would not have been able to give this presentation today. And then, of course, the global public health community of pathogen genomic scientists, and especially the public health scientists hosting this call today with PGC Mindanao. Thank you all very much. I will take some questions now.
Thank you very much, Frank. That was a very illuminating talk. I can say on a personal level.
I am jumping into the question and answer box, and I'm going to go in order from top to bottom, as long as that is okay with our host. Carlo, does that sound good?
Yeah, sure. If you don't mind, I actually wanted to highlight.
Okay, so
go ahead.
Let me see. I think I'm going to turn on speakers. Okay. All right. Can you hear me, Carlo? We're all set?
Yes. Can you hear me?
Okay. Yes, I can. Thank you.
Okay. Good. Yeah, go ahead. Yeah, the first question, if you don't mind, I'd like to highlight the ones that we'll be addressing. Personally, I have my own, a lot of comments and insights I gathered from your talk that would be interesting to discuss. But let's start with the second and third questions are both relating to what I imagine are beginner bioinformaticians. And this is a going concern because I deal with training bioinformaticians in the Philippines. And I think what I've observed is that a lot of people are public health professionals, doctors, nurses, laboratory technicians, medical researchers, whose background is not really into bioinformatics at all. They don't know how to code. Maybe even their knowledge of next generation sequencing is limited. And then the pandemic has really highlighted that, just like you've discussed in the talk, bioinformatics and genomics are really powerful tools for dealing with public health concerns, something that might not have been they haven't been very conscious of before. So now we're seeing a lot of interest of people who want to shift or add to their careers and develop their bioinformatics skills. So the second and third questions here, I think, relate to that overall concern. What would you tell somebody in that situation? What skills would they want to develop, or what message do you have for them to get into bioinformatics when they're basically starting from an entirely different field?
Yeah, it was not long ago that I had these same questions myself. So I remember the idea of trying to learn all of the coding languages is very daunting. And even picking one, it feels like it's not going to be enough. And I fully understand and appreciate that challenge and that question. So thank you for these questions. I would say the most important skills that you want to learn are not how to code, and it's not how to implement bioinformatics algorithms. I would say the most important skill that you need to learn is asking good questions, trying to figure out what do you want to learn from DNA sequencing, whether it be how do I detect antimicrobial resistance in a pathogen? Now, that question is very general, but if you can learn to focus your questions to how can I detect an antimicrobial resistance gene, or how can I detect an antimicrobial resistance conferring point mutation? So these are the types of questions that are easier to answer than just the general, how do I detect antimicrobial resistance? I would say you don't need to learn how to code because there are so many bioinformatics analysis softwares out there that are accessible through graphical user interfaces. So that's where I would start, is go find the tools that you don't need to write code to perform the analysis, and just start performing the analysis. Look at the inputs, look at the outputs. You don't need to learn exactly how the machine works. If you see the inputs and you see the outputs and you start to understand what the outputs look like based on the inputs, then once you get a feel for it, you can start to figure out, okay, how do I change things? How do I change certain parameters? Not everyone needs to build the machine. Some people just use the machine, right? So in my mind, there's many different areas for bioinformaticians to exist in. Some of them will be development, some of them will be implementing the code, but some of them will just be using the code that is out there already, whether it be on the command line or behind a graphical user interface. So my advice would be just start doing the analysis with whatever tools you can use. And I would start with anything behind a graphical user interface first.
Okay, thank you very much. I have to admit, I have a different philosophy from you because I grew up as a bioinformatician in what we might think of now as the old school style. So I did learn to code, I learned how to use Bash, and so on. And one of the first things I tell people is, I guess I expect them to mirror my journey. I tell them, yeah, pick up a programming language. So I really value the insight now that, honestly, I have this vague sense from listening to your talk that things are shifting. Maybe we're being left behind, and that there are these new tools and platforms out there that we have to adapt to. So I really appreciate that. And then the topic of cloud-based services, I want to highlight this question near the end because they had a similar concern. So they ask that they're concerned about cloud-derived data used in informed decision by authorities. So how do they know the data that they have stored and retrieved in the cloud are safe or true data? And actually, it's the next question that I think is more relevant. Who owns and controls this data? And if you don't mind my adding to that a bit, because people working in a public health context will of course have to deal with private medical information, personal patient information, private genetic information, and of course there are a lot of concerns about protecting that ownership and privacy. The Philippines itself has stringent laws when it comes to data ownership and privacy, and we don't like to give our data to the control of other countries or institutions because, as a developing nation, we're prone to that exploitation. One of the reasons my institution, the Philippine Genome Center, exists is to provide services to Filipinos without having them go to other parties. So I guess, so that's the overall question, I guess. How is that concern addressed when it comes to ownership and privacy with cloud-based services?
Yeah, that is a fantastic question that I have received many times. And the best answer I can give is you own your cloud repository in the same way that you own your hard drive locally. And you are you have decision-making power over what information gets shared, who has access to that information. And these cloud providers, they tend to put the onus on you. There's a split security model where you are in charge of who has access to the data. They are in charge of security over the physical hard drives themselves. Now, they're never going to just pull data out of your hard drive and go and share it or sell it. That is built into the contracts when you sign up for cloud. And I know it feels scary to send your information to another computer and you don't know where that computer is necessarily. You can't go turn the computer off if you want. But in my mind, I've gotten so comfortable with it at this point that it is essentially like having your own computer. When I access the cloud through my computer, I think of it almost as if it is my computer.
Okay, thank you very much. Yeah, I hope that's reassuring to the people who have those concerns. Let's tackle this other question on the type of targets that tend to be used in biosurveillance. So somebody asked, is sequencing and assembling eukaryotic pathogen genomes for biosurveillance becoming more common? Because of course, eukaryotic genomes are going to be significantly larger and more complex than viruses. Even bacteria, larger eukaryotes would have larger genomes. So is routine biosurveillance for eukaryotic pathogens feasible in the near future?
It's actually very serendipitous that you would ask this question right now, because there is currently a Candida auris outbreak in the Pacific Northwest of the United States, and we are performing outbreak investigation and genomic surveillance on this eukaryotic pathogen outbreak. Now, yes, the genomes are much larger, and yes, the resistance is different, but so Candida auris is a fungal pathogen that is inherently resistant to many antifungals. So antifungal resistance detection is extremely important. So now you're not just doing assembly, but you're doing and quality control, you're doing assembly, quality control, antifungal resistance detection, and eventually you want to be able to do genomic epidemiology. So you want to place all of these genomes in a phylogenetic context. And that's where it gets a little bit more tricky computationally, because creating a phylogenetic tree, it scales non-linearly with the sizes of the genomes. So the bigger the genomes are, the more computationally intensive, exponentially more computationally intensive building the tree will be. So in my experience, that's where you need to get creative with your pairwise comparisons. So for example, if you're trying to compare 100 Candida auris genomes against one another, you can do it with a variety of tree building tools. And we're actually in the process of doing this right now with a tool called 𝗘𝘃cie𝗹𝘂𝘁. We're doing pairwise SNP analysis between these genomes. And just for context, the Candida auris genome is about 12.5 megabases. So not that much larger than your typical bacterial genome, probably like twice as big or 3 times as big as your typical bacterial genome. But to answer your question more succinctly, as we start analyzing more eukaryotic genomes, we will have to get more creative with our phylogenetics specifically. As far as assembly and antimicrobial or antifungal resistance detection, I think we're already there. And with compute technologies in the cloud being virtually infinite, in my opinion, all you need to do is add a few more CPUs and you can get there in no time.
Okay, thank you very much. Yeah, I'm glad you had such an apt response to this question. Let's tackle this question by 𝗘𝗃𝗃𝗁𝗃𝗃𝗃𝗁𝗃. We've seen a decline in malaria cases in the Philippines in the last decade, with only 4 provinces endemic for the disease without relying on molecular methods for diagnosis. Realistically speaking, we always resort to the less specific routine methods due to the lack of funds to support these highly sensitive methods. I think this speaks to my earlier mention about how resources and like advanced technologies aren't always readily applied here in the Philippines, right? So they ask, how do researchers or people in your locality encourage your government or funding institutions to fund omics diagnosis of other endemic diseases? Yeah, if you'll find that dealing with Filipinos, funding concerns are always prevalent. We're always searching for ways to secure funding. So, do you have any insights on that topic?
Yes, absolutely. I have worked in several laboratories where funding was an issue. And the number one argument I've heard for using omics tools is that most assays are very specific. They're often pathogen specific. Unless you get into like a real-time PCR machine, you might be able to expand it, but those assays are very specific. DNA sequencing is not specific at all. You can perform DNA sequencing on any pathogen. So when it comes to funding for a sequencer or sequencing reagents, my argument is always, you don't know what the next public health pathogen outbreak is going to be. And because you don't know what it's going to be, it's probably best to invest in the most generalizable technology. Additionally, sequencing technology can give you the most information per analysis. So if you're just performing PCR, you might be able to determine whether or not there's a particular genomic feature present, but DNA sequencing will allow you to get all of the genomic features present. So if there's some novel form of antimicrobial resistance present in your samples, you will detect that with sequencing and you will not detect that with any other assay. So those two arguments, the fact that it's highly generalizable and the fact that you get the most information possible out of each analysis.
Okay, thank you very much. I hope that answers the question. I'd like to insert one of my own questions, if people don't mind. Apologies to our audience. Listening to your talk as somebody with some bioinformatics experience, so I'm curious about, but I don't really have any experience with cloud-based platforms like 𝗘𝘃cie, like you mentioned. So I'm curious about that. So you describe it as having a graphical user interface, which leads me to wonder, how much fine control would the bioinformatician have in developing their workflow? Because the nice thing with scripting is more complicated. You have to know how to program, but you can control everything if you're the one scripting. You can troubleshoot. You could refine your pipeline exactly the way you want. Pump in the maybe specific metadata or produce whatever exact specific outputs you want. How does one, and with a graphical user interface, it's easier, but maybe you have less control. So how does that trade-off happen on a cloud-based graphical platform like 𝗘𝘃cie?
That's a very important question. So there's really two sides to this. First of all, when you develop a workflow on 𝗘𝘃cie, you can expose any of the input parameters to your tool. So when you're running a command on the command line, you'll know this, Carlo, but there's often flags that allow you to assign particular inputs. For example, the minimum depth of coverage at a nucleotide position that you need in order to assign a base when assembling a genome. You need to have 100x coverage per nucleotide. Now that input parameter may or may not be exposed in a graphical user interface. You may or may not have access to that. Now, one of the best practices that we use at Theogen Genomics is to try to expose as many, if not all, of the input parameters for each component of the pipeline. So your read trimming parameters, your read QC parameters, your assembly parameters, your antimicrobial resistance detection parameters will all be there. Now, the trade-off with that is you can end up with 100 input parameters available in your graphical user interface. Now, we set intelligent defaults, meaning you can just enter the 3 required inputs of your read 1, your read 2, and your reference genome, or whatever it might be for a particular workflow. But you have access to all of those dials and switches in the graphical user interface. They have defaults set, so you don't have to play with them. But if you are concerned about a particular input, you can adjust that. Now, on the development side, because 𝗘𝘃cie is an open source platform, and because all of our code is open source, if there is a parameter that it's not exposed that you would like to have access to, you can actually modify our script on your own development branch and make that available on the graphical user interface so that you have access to every input parameter that you want personally.
Okay, thanks. I didn't know that, so that's very useful to know. There's this other question in the Q&A box. This might, I think we need to wrap up. So I think I'll just address, actually the first question in Q&A. I think it's a good way to close this, this webinar. So there's this very enthusiastic question. They say, the evolution of sequencing technology in bioinformatics is really amazing. So where do you think bioinformatics could lead to in the near future in terms of public health or disease detection?
Oh, this is my favorite question. I talk about this question with my friends all the time. And I foresee a future where the entire global public health community is all working together. And we have formed a sort of digital immune system where all sequencing data is being submitted to repositories such as NCBI, and we all have access to each other's code, and the development is so collaborative that every bug is worked out immediately and all of our pipelines run perfectly, and everything is analyzed automatically upon submission to the repositories, and alerts are sent out automatically if any public health pathogen of concern or antimicrobial resistance is detected.
Okay, thank you very much. I think that's pretty much all the time we have. So personally, I want to thank you. I genuinely found your talk illuminating and insightful. And now I'd like to turn the floor back over to our hosts to wrap up this webinar.
Right. Thank you for that wonderful talk and for the engaging question and answer portion, Mr. Ambrosio and Mr. Lapid. We regret that we don't have enough time to answer all of your questions as much we would like to. However, don't fret. For the other questions not read in this session, or if you have any other questions in your mind, you may contact our speaker or through the PGC Mindanao email address so that we can relay your question to the speaker. To all our participants who will be needing a certificate of participation for this event, please wait until the end of the webinar to access the link to our evaluation form, in which we will post it in the chat box. A certificate of participation will be instantly sent to your registered email address when the evaluation form has been successfully submitted and evaluated. At this juncture, we would like to give our appreciation to our moderator and speaker. So I would like to request our moderator, Sir Carlo Lapid, and our speaker, Mr. Frank Ambrosio, to turn on their cameras. And we also would like to request the PGC Mindanao Director, Dr. Tolentino, to turn on his camera as well. Right. Yeah.
Hello.
Yeah. So let me read the citation. A certificate of appreciation is proudly presented to Mr. Frank Ambrosio for being the resource speaker on the evolution of public health pathogen genomics during the Bioinformatics lecture series held on November 21, 2022, signed by 𝗘𝗃𝗂𝗁𝗃𝗂𝗁𝗃, PhD, Technical Expert of the Bioinformatics Division, and to our signed also by Dr. 𝗃𝗃𝗁𝗃𝗂𝗁𝗃 Tolentino, the Program Director of PGC Mindanao. Thank you very much, Mr. Frank Ambrosio.
Thank you all very, very much. I really appreciate it. Thank you so much to the hosts, and thank you to PGC Mindanao for having me. It was an honor and a pleasure to present for you today.
Dr. Frank.
Thank you, sir. Also, we would like to give this certificate of appreciation to Mr. Carlo Lapid for being the moderator during the open forum of the talk entitled The Evolution of Public Health Pathogen Genomics by Mr. Frank Ambrosio in the Bioinformatics lecture series held on November 21, 2022, signed by Dr. 𝗃𝗃𝗂𝗁𝗃𝗂𝗁𝗃, Technical Expert Bioinformatics, and Dr. 𝗃𝗃𝗁𝗃𝗂𝗁𝗃 Tolentino, Program Director of PGC Mindanao. Thank you very much, Mr. Carlo.
Thank you very much as well. It was an honor and a pleasure. And thank you, Frank, for your talk.
Thank you, Carlo.
Once again, thank you to both of you. At this moment, I would like to request that you keep your cameras open for a photo opportunity. So please tidy yourself up and show us your best smile. And one of our staff will give us a cue for picture taking.
All right. 1, 2, 3, smile.
Yes, sir.
Okay,
Okay.
na.
Okay, thank you, Dr. Tolentino, Mr. Ambrosio, and Mr. Lapid. We will also would like to request for the rest of the organizers of this event to turn on your cameras for a photo opportunity. I'd like to call on the rest of the Bioinformatics Division staff to open your cameras. And again, one of our staff will give us a cue for picture taking. And
All right. 1, 2, 3, smile. All right, one more time. 1, 2, 3, smile.
yes, sir. Okay, right. Thank you for that. Now, at this juncture, we would like to hear from our dear participants on how we did in today's event. Our team will give you the link to the evaluation form in the chat box, and before the end of the day, kindly complete them. And once the survey is turned in, all participants will get their certificate of attendance. So the link is already in the chat box. And in the event that you do not receive your certificates within an hour, please email us at pgc.upmean@up.edu.ph. Okay. So for future events and to learn more about the services that we offer, please visit our website at pgc.upmean.edu.ph, as well as our different social media accounts like Facebook, YouTube, Instagram, and Twitter. And you may also contact us through the number shown or email us through pgc.upmean@up.edu.ph. And that wraps up our third lecture for this year's lecture series on bioinformatics. Next Monday, November 28th, we will be having Mr. 𝗃𝗃𝗁𝗃𝗃, a CEO of 𝗣𝘂𝗃𝗃𝗁𝗃, to talk about the bioinformatics in applied microbiology, the last remaining barrier, as our last speaker for the Bioinformatics Lecture Series 2022. We hope that you can join us again on November 28th. The registration link for the webinar will be posted by one of our staff members this week. And please register by clicking the link and reserving your spot. Again, on behalf of the Bioinformatics Division, I'd like to thank you for attending the third lecture of the Bioinformatics Lecture Series 2022 of PGC Mindanao. We wish you everyone a good day and ask that you all be safe. Bye everyone and see you next week.